Good evening to one and all. I hope I'm audible. Can you hear me?
Yes, sir.
Wonderful. It's a delightful evening, an hour to be spent with young people, hopefully both from the faculty and the students, not only from St. Stephen's College, but other places as well. Our talk today is entitled Fullarazirman. Our talk is entitled Fullarazirman. Even if I succeed in this 1 hour to explain just one word, the new word that you have heard from me, coined by me and available in literature since 2020. I repeat, Fullarazirman. I wonder if any of you have heard of it before. We go on to the discussion by moving to the next slide. This is a summary of what I said last time. I will only spend a word or two on this and move on to the main talk proper. And that is, what is the purpose of doing science in a college? Is it that we have to do research like in the national laboratories, National Chemical Laboratory, National Physical Laboratory, and so many CSIR laboratories there are in this country? Or is it the purpose that we do research as a hobby, along with teaching duties and administrative duties which come to teachers, and most importantly, the mentoring part, which you will see very often absent in many organizations, including the university and the industrial structure. I'm not going to elaborate on this point, so we can go on to our next slide. And I think these are some questions which have been raised, which were raised by me as early as, say, 2005 or 2004. And I went to the National Physical Laboratory in Pusa Institute. We actually published a paper in 2011 with the scientists from the National Physical Laboratory of NPL on solar cells being made with similar materials. That was in synthetic metals. Those who want to read the paper, go to the journal Synthetic Metals. Maybe I asked one of you, maybe Prajushya, to raise the questions, to which I will give brief answer.
Yeah, the first question is, what is fullerene? In brackets, Buckminster fullerene.
I think we all have seen this molecule which looks like football with hexagons and pentagons, but contain number of double bonds. The shape is that of a football. I never had Buckminster shape shown to me, but I have had the sample. This is a nanocarbon. It is a new allotrope of carbon, which was synthesized successfully in the 1980s, for which the 1985 Nobel Prize was given to 3 people. It is like a Buckminster dome, which has no pillars. Fullerene is nothing but Buckminster, fullerene. It has 60 carbon atoms. And those who are students of chemistry, I presume most of you are students of chemistry, we know the atomic number and atomic weight of carbon. So the atomic weight is 12, standard weight, taken as a standard. And if there are 60 carbon atoms, is very important for this lecture. The molecular weight of fullerene, C60, must be 720. Is that correct? Have you understood me? What is the molecular weight of fullerene? 720. Did I make fullerene? I have never claimed I have made fullerene. I bought fullerene. People in India are now making some fullerene. At that time, when I bought it in 2010, or I forget when, 1 gram of fullerene cost me 52,000 rupees. What is the next question, my friend?
What is the potential and limitations of nanocarbon materials?
The potential, people say, is unlimited. There is a young man who's maybe approaching 40 now, Gadadhar Reddy. There are videos of him in the internet. And believe me, what is interesting is that he has made another nanocarbon material in collaboration with Jet Propulsion Laboratory, student of a Nobel laureate. And his company is known as PO, No Pore Technologies, not possible technologies. He makes very ultra pure single walled carbon nanotube, yet another allotrope of carbon, but not yet decorated with a Nobel Prize. There is a third one you will hear briefly, graphene, which won the 2010 Nobel Prize. What is the potential? People talk so highly of these materials, but they're costly, prohibitively costly. And more important, they're totally insoluble. Whether it is fullerene C60, fullerene C70, single-walled carbon nanotube, multi-walled carbon nanotube, or graphene or reduced graphene oxide, to which we'll make a reference in this talk, they are all black in color, totally insoluble in most solvents. What do you do with the sugar which you want to add to your coffee or tea, but which does not dissolve in your tea or coffee or water? How many such materials? I told you just, I mentioned 3. So I don't have to repeat, but I'll just mention all right. Fullerene C60, single walled carbon nanotube on which I may present something today. Graphene, which I could not lay my hands on yet. Do these increase lifespan of human beings? Sir, are you giving a lecture on physiology and medicine, or you are giving a lecture on chemistry/science? Excuse me, whole of America, without taking any advice from any doctor, they are consuming fullerene in olive oil because they believe they can live longer. Deepak Chopra, who is my contemporary, but is much, much, much richer than me because each lecture of his very costly to attend. His books are so popular. He's a very popular author and a speaker in America, a MBBS from All India Institute of Medical Sciences, my batch. People like him tell now the 125th birthday or 150th birthday will be celebrated very ordinary manner as if it's nothing special. People now celebrate their 60th birthday, 70th, 80th birthday. That'll all be a thing of the past. Whole of America and many places believe that human lifespan can be increased by consuming this nanocarbon material. Yes, what is the mechanism of action? It appears that they are all radical scavengers, oxygen scavengers. That means they take away free radicals formed in our body, and free radicals play havoc with our body. Many doctors now routinely prescribe what is called, antioxidants. They also work in the same manner. So one can remain more healthy. All right. You are saying they are totally insoluble, but you are now mentioning that there may be water-soluble nanocarbon materials. And their importance, I told you, something soluble can be injected, something soluble can be taken orally, something soluble can be, given as an intravenous injection. One of the main purposes of this lecture is to tell you what is Fullarazirman and why I named it thus. As we go to the next topic in this talk, instead of my saying this, our person who is helping me will move to the next slide. Hi. Is that Buckminster football or fullerene on the head of whom? This is in a, big ship. I don't know what you call such big ships, where this Nobel laureate Croto, W. Croto, is holding a balloon made Buckminster fullerene, of which he is one of the discoverers. He shared the Nobel Prize 1985. What does he advise young people? He says, with this recipe, whatever your limitations, you will almost certainly still do better than anyone else. What is the recipe? Tell me, read somebody. It is If it interests you, however mundane it might seem on the surface, still explore it. You think it's very ordinary, still explore it. Something unexpected often turns out just when you think you least expected it. He says, don't go after awards because you will have disappointed. He says, who are the people who are most interested in science? He says, young people and retired scientists or retired people. This is not my comment. This is Kroto's comment. Actually, my wife and we were on the ship in Lindau, in the Lindau Nobel Prize winning meeting. I was there for the second time, invited by the Germans. Suddenly she said, come with your camera, come with your camera. I saw Kroto holding this on his head. This is a photograph taken by me on the ship between Lindau and Mainau, where every year there's a Lindau Nobel Prize winners meeting held. We go on to the topic that we discussed today, Fullarazirman. Can you see that the three allotropes of carbon on the left-hand side is a football-like figure. In the middle is a tube-like structure. Actually, it rotates. This is not a slide view where we can see the rotation, and a flat two-dimensional surface of graphene. These are supposed to be stronger than steel. But since they are totally insoluble, most people treat them with concentrated nitric acid and concentrated sulfuric acid. The closest I have come to is an H.G. Wells movie where a man was thrown into the bath of boiling concentrated sulfuric acid. I mean, why do you treat such things with such strange and such strong reagents which may damage their critical structure of the nanocarbon material? These are the three materials that I have mentioned to you. Graphene, single-walled carbon nanotube. Of course, there is the multi-walled as well. And graphene, two-dimensional, single layer, double layer, and so on. Yes, this is what is the definition of nanocarbon materials. You must move on to this next slide. Don't expect me to say this again and again. Then it interrupts the talk. How do you functionalize? If you functionalize, two advantages come to us. One advantage is that the toxicity of these materials disappears because they are nano size. They can enter into your lungs and produce toxicity. But if you functionalize, do some reaction like a Bingle reaction or a Prato reaction. Bingle reaction is the carbene intermediate to which I made a reference in the first lecture, and Prato reaction is with the nitrine to which also a mention was made. Therefore, you must understand all the 11 lectures have a common thread. The common thread is I went to Germany in 1984, but the work I did in Germany in 1976-77 started my interest in nitrines. Then I realized that I have met Professor Platz in Ohio State University, who is an expert on aromatic azides and azides in general, and particularly on carbenes and their physical chemistry and their energetics and laser flash photolysis. I was with him for some time, and he's such a remarkable figure in the sense he used to eat a lot. Now he's thinned down because he's taken care about his diet. Prato reaction with nitrines and Bingle reaction with carbenes. Or you can just have covalent interactions for functionalization. Functionalization makes these substances soluble. If they're soluble, they can be processed. Another reaction is cycloaddition, and the one that I mentioned in the previous slide, that is using concentrated sulfuric acid, nitric acid, and heating the nanocarbon material. This is a very harsh method. We don't do this. We are gentler Indians. Yes. There is an article of mine which is being cited highly. I recommend this entire audience to read my article entitled Water-Soluble Nanocarbon Materials, Water-Soluble Nanocarbon Materials, a panacea for all, whether it's cancer, whether it's tumor, it's arthritis, Parkinson's, HIV, AIDS, you name the disease, it appears that water-soluble nanocarbon materials are able to treat all these diseases. Is it an elixir for life? I do not know. Please don't get my point take it too seriously. What I'm saying is I'm not commercially selling any idea. I'm saying this is what people are believing. Literature is showing papers. Every single sentence here is supported by many, many research papers. You can get them on the internet yourself. They are saying breast cancer can be treated. And the amounts of these nano substances as drugs that is needed is very small. So the side effects will be very, very small. And if they are water soluble, they will not stay in the body. They will be just passing out. We go to our next point. Young man, smoothly move on. This is a photograph from my article in Current Science 2016. Current Science is a journal published by the Indian Academy of Sciences. And here it shows how a tumor in a rat was actually treated in this manner. So I do repeat, fullerene drink might be an elixir for life. हो सकता है अमृत हो. देखिए, जादू जो होता है जिंदगी में कोई जादू नहीं है. There is no magic. Hard work, dedicated work, huge amount of reading, and reading between the lines. I had started reading, as I said, 2004, 2005. This is a paper published by me in 2020, a very highly impact factor journal, very costly also, but it's an open access journal. I'll read the title, Fullarazirman. Oh, acha, this is not a name unknown to science. It is in this journal of biomedical nanotechnology. But if you have to access this paper, you have to shell out, I don't know how many pounds or dollars. Open access, you can get the abstract. You can't get the paper. You write to me, I'll send you the free paper. Fullarazirman, nano water soluble fullerene C60 manganese tri-adduct complex. Yep. Who is it? Hello? Yes? Triadic complex with anti-cancer activity comparable to that of doxorubicin, the currently most used anti-cancer drug. But this drug has so many side effects that the patient dies not of cancer, the patient dies of heart attack. So how do we get a drug which can be used in lower doses, less toxicity, higher activity, and yet much safer? One of you can ask me a question at the end of this talk or during the talk. You can put it in the chat box. The question answers will be taken up. Each of the questions will be answered. If not, we will be in touch at the end of the lecture. You can approach me. Am I claiming something extraordinary? Hard work. Every reference given in this paper I have read. You have to read the paper many, many times to understand it. Even my paper is not kid stuff. Even if I send you the paper, you'd find it difficult to understand it. That is the need for this lecture. Yes? See, when we made it, it took a lot of effort. We made a reaction between fullerene C60 and the nitrine reactive intermediate from azido-metameconine, and also azido-metahemipinate, from which bisphomyine was formed. But this product is by heating in two steps, fullerene C60 with azido-metameconine. Aryl azides are today known as green reagents. Why? Because when you heat them or put them in the sun or UV light, they lose nitrogen. They are therefore only producing the innocuous nitrogen, which does not pollute. So they are called as green reagents. One of my students from St. Stephen's College went on an Erasmus scholarship around 10, 12 years ago, and she has published a paper in Journal of American Chemical Society. It's a very prestigious journal, and she's the first author. She did a PhD from Holland, and she has described there that aryl azides are known as green reagents. What is this slide showing? This is called MALDI MS. I moved away from St. Stephen's College post-retirement in the year 2014. I was an emeritus scientist, the only one in the country between the period 2012 and 2014. But for whatever reasons, someday we will discuss this in person. I had to leave St. Stephen's College, where my heart resides even today. हमें तो दीवार में चुना दिया गया है भैया. दीवार के चुने हुए लोग कहीं भी हों, दिल तो वही है. आप समझते हैं इस बात को? पता नहीं कितना समझ पाएंगे. आज से 50, 60, 70 साल बाद you may also understand the intensity of my statement. शायद, maybe you find it difficult to read the last value on the right-hand side, but I'll tell you, it is written at the bottom that the value is around 1492.9912. So the fourth decimal place, kitna value hai? 1492.9912. I told you in the beginning, what is the molecular weight of fullerene? 720. And my reactive intermediate, the nitrine from azidometameconine, has an atomic mass unit of 2. Yes? Yes?
Sir, I am Saptarshi. I have one question, sir. Sir, you have...
my young friend, can you wait? Yes, ask the question, please, briefly.
Sir, my question is, what is the use of...
Yeah, we understand his question. We will help him get the answer. He's going to get a big, big answer shortly. Are you hearing me, all of you? Go ahead. 1493, 720, you add 239 multiplied by 3. 3 of my units in a symmetrical fashion added to 720. 239 into 3 is 717. 717 plus 720 is 1437 or so. But it is nowhere near 1492.9, which is almost 1493. I guess that the difference of 55 is the exact atomic weight of one manganese atom. One manganese atom is 55. So if you add 55, you get the molecular weight of this new compound, which is now named as Fullarazirman by me. I hope I repeat. Molecular weight of 720 of fullerene C60, 60 into 12. 239 is the molecular weight of the species called the nitrine from my azidometameconine, which takes a lot of time and effort to make it. It's our compound described. Nobody else has described this in literature before. Made in St. Stephen's College when I was active. Then I had to move to a regional center for biotechnology. There was this multi-MS instrument. Suddenly God threw me out to a place where most sophisticated instruments are available. My student got the spectrum, 1493. Sujit is here with the excitement of this entire work. I know. I knew for certain there must be a manganese atom. There is some disturbance. हम सारे क्वेश्चंस आंसर करेंगे, कोई क्वेश्चन नहीं छोड़ेंगे, पर पहला पूरा हाइपोथेसिस सुन लीजिए. 1493, 1493 is possible only if there is a single manganese atom. How does one detect manganese in St. Stephen's College or any college? We do the analysis in 4th group. We do the analysis and we get to go to the 4th group and finally get a pinkish precipitate in the 4th group. As we go, the question was asked, how to prove presence of manganese by a modern technique. Can you believe it? As expected, we went to the JNU, Jawaharlal Nehru University Instrumentation Centre. And recorded an electron spin resonance spectrum. Manganese is known to give a Manganese is known to give us 6 lines of equal intensity, parallel to each other. We got 6 lines. I hope all is well. We got 6 lines as expected, but they were not symmetrical parallel lines. It's an asymmetrical spectrum. All the lines were of different intensity. And there was a bunch somewhere down on the left-hand side. So it was very clear that manganese is present in this molecule. Next slide. The various values of the ESR spectrum are shown in this slide. Anybody wants to note these, we can give you subsequently as we go to see the picture of the molecule itself. Yep. We go to the next slide. See, to prove that it is in fact nano, we did the zeta potential measurement and the size came out to be 58 nanometers or so, which is well below 100 nanometers. Fullerene C60 is even smaller, but 3 units of my aryl azide nitrine got added along with the manganese. That means somewhere somebody has to switch off his mic. Please switch off your mic, because we are hearing background noise. Somewhere it is raining, it seems.
Sir, it's done.
Yeah, done. Now, see, what we imagined was there is one fullerene, 3 units of the nitrine from azidometameconine added in a very symmetrical fashion, and then a manganese atom on right on top of it. Manganese on the outside or manganese inside the fullerene. When it goes inside the fullerene, it's called endohedral. This is outside the fullerene ring, and the complex was thus in my sight. Next slide shows you the sample. What a beautiful sample. The photograph is taken by me. This sample does not exist anywhere else in the world. It should be put up in a museum of St. Stephen's College, science contributions. 100 years later, one would come and see the sample. I went to Germany, Göttingen. I saw a museum in the university chemistry department where the original sample of urea made by Friedrich Wöhler was kept in such a test tube. They have preserved the sample. In India, I do not know we have such strong traditions of musea, especially for science and what happens in the college. But I think our college will learn this someday better than others. We have a tradition of a strong alumni, and at the same time, we have the tradition of preserving our various traditional items. Like, for example, some of you may not know what is the Rudra dinner. Rudra dinner is held on the same day, same night, I should say, every year, 12th of February, if I remember right. It is a dinner organized by none other than Dinabandhu C.F. Andrews, Charles Freer Andrews, for his friend, the Principal S.K. Rudra, whose birthday it was, if I remember right. On that night, I claim I have made 6,000 puris every year. You go to the kitchen, you make puris and serve this to along with aloo ka sabji and so many other things to the workers. All the workers and their families come and eat in a very joint, happy fashion. Then they do some drama, theater, and so on. Some of them act in a drunken manner. I don't know whether they are actually drunk or not, but that is the tradition. This photograph should belong to the museum in St. Stephen's College. This is a photograph, Fullarazirman, named by me, it's water soluble. Light pink in color, shows possibly the presence of manganese. The next slide will show you. Nano substances are known to form flower-like structures. Can you believe it? Under the scanning electron microscope, we took a photograph of this substance of ours. It is just like a carnation flower. I do not know why such flower-like structures are observed when you have high-resolution microscopy done on these samples. I'm sure everybody present in this audience or this group, I do not know how many people are present, how many are enjoying this. This is a spectacular photograph. Makes me extremely happy. I'm not saying it makes me proud. Why should I not be proud? Molecule made by my team, with my workers. How many workers? One worker. Was he paid heavily? No. Was I paid heavily? No. Did we have places to work? Don't talk about these things. Work is more important. Comes in front of us. Remarkable photograph confirming its nanostructure. Next. Now the cat will be out of the bag soon. Can you see Fullarazirman? Students of chemistry are finally seeing a structure. What is in the middle? Fullerene, C60. I understand my student is asking, what is its use? I'm going to explain shortly in the next few minutes, the second part of the talk. Can you see Fullerene C60? No, go back. On 3 aromatic rings are 3 nitrogens. They are forming a 3-membered aziridine ring with the fullerene double bonds. How many units? 3 units. The free carboxylic groups, there are 6 of them. Only 3 of them, the carboxylic group away from the nitrogen, the one that is near the nitrogen is blocking or is blocked or sterically hindered, or we call it even, with other words, it is actually not allowed to participate. It is an introverted carboxylic group. There are students who are introverted. They are hesitant to talk to each other. But being introverted, I'm told, sometimes leads to dementia. So it's better to be, we are all psychosocial animals. Man is a psychosocial animal. So it's better to be communicating with each other, sharing one's joy and sorrows. I told St. Stephen's greatest tradition is the fellowship on the campus. People live together as a family. People visit each other on Diwali. People visit each other on Christmas. Christmas carols at midnight and the carol service in the college hall are all such delights in such cold, freezing conditions which can happen on the 25th of December each year. What is Fullarazirman? It is a manganese complex of fullerene C60, aryl aziridino manganese 2 complex. Full stands for fullerene. AR stands for arene, aromatic, aryl. Full stands for fullerene. R stands for aryl. AcSIR, AZIR stands for the 3-membered aziridine ring, 3 of them, and MAN stands for manganese. So I gave the name Fullarazirman. The name was eventually published in Journal of Biomedical Nanotechnology, as I said earlier, in 2020. Next slide shows you. Next slide shows you the tremendous areas of interest that could be there in this materials of this kind. This comes very close to the question Saptarishi likes to raise. What is its use? You can make a transistor, you can make solar cells, you can make energy storage devices, you can make, for that matter, things and use it for gene therapy, you can use it in drug delivery, you can make biosensors, among many, many, many uses that one can think. I hope you have understood in this first part, major part of the talk, what is Fullarazirman. Fullarazirman is a molecule born post my departure from St. Stephen's College, strictly speaking, in the Regional Center for Biotechnology of the Department of Biotechnology in Faridabad, a state-of-the-art laboratory with most sophisticated instruments. If I did not have access to the MALDI, that is matrix-assisted laser desorption, ionization, mass spectrometry, I would not have been able to record the molecular weight of 1499, 92.99, etc., whatever you saw, that is 1493. Had I not been able to record, I would not have been to show the presence of manganese. And manganese formed a complex with 3 of the carboxylic groups. My student, Dr. Sujit Kumar Thakur, who is present in this group, I presume is present. He knows how many times we have had discussions, how to establish the presence of manganese. Take it to St. Stephen's College, third-year honors lab, and do some sodium fusion or say sodium carbonate solution preparation, and then do the 4th group analysis or other methods by say sodium bismuthate and get a pink color, blah, blah, blah. But we didn't do it. We did it by electron spin resonance spectroscopy, ESR, in Jawaharlal Nehru University. Fullerene, R, aryl ring, 3-membered nitrogen ring, AcSIR, manganese substituted or reduced to MAN, AMAN, gives the name Fullarazirman. But this is water soluble. Did we stop there? You see, when you get an idea, you get a method, don't stop there. Somebody in this country who's very famous, you have a very good quotation, arise, awake, and stop not till the goal is reached. उठो जागो पर अपने ध्येय को पहुंचने तक रुको मत, चलो काम करो. At this age, I'm prepared to talk into midnight, but we have to close this lecture within the period of an hour and also give time to young people to ask questions. Next slide shows. We have done the same work with single wall carbon nanotube. Again, this was white in color. Again, we did studies on plants, and we showed that the root length increases in Arabidopsis thaliana. Arabidopsis thaliana is one of the 3 model systems in which they test new materials for use in human beings. The other is Drosophila and so on. So Arabidopsis thaliana was available to us. And if on the left-hand side, you can see the expansion. Huh?
Sir, is it Drosophila melanogaster?
Yes, that is another. We have not done that. That also work we have done. We have published separately on IP6. There are 3 systems like this. One is the Arabidopsis thaliana, then there is melanogaster, Drosophila, the fruit fly, and there is one more very transparent material, insect, on which they test this new materials because their genes are Drosophila that was mentioned is very similar to human genes, except the number of genes is very small. But whatever results you get, you can carry over to human beings. Next slide. This work was taken further by my colleague, Dr. Prasanjit, who is senior most professor in Regional Center for Biotechnology. And in his lab, they showed that, you see, Fullarazirman is very, very effective in this immunoblot on the right-hand side. It shows that shows the efficient apoptosis. Apoptosis is the cell, programmed cell death inducing ability in monocytic U937 cell lines, which are cancer cell lines, when they are treated either with our compound Fullarazirman or with doxorubicin. The results are similar. Maybe our compound may be better because the cytotoxicity of our compound is very, very small. Cytotoxicity of our compound is very, very low. So our compound may be more safe. These tests were done at different concentrations, as you see in the bottom of the bar diagram on the left. That is at different concentrations of 6.2 micromoles, 3, and so on. And the results are very, very good, right? So, and these have been tested in CT26 cell lines. This work is done not by me, not by my direct student, but a student of my collaborator, Professor Prasanjit Guchayat, who works in the Regional Center for Biotechnology. Next slide. If you want more explanation, Dr. Sujit Thakur will explain this slide to anybody who's interested in this. But we moved away from Fullerene, Fullarazirman, single-walled carbon nanotube. We bought reduced graphene oxide from Mohali. A company in Mohali is making. 1 gram cost us only 4,000 rupees. Damn cheap compared to 52,000 rupees for a gram of Fullerene, 52,000 rupees for a gram of single-walled carbon nanotube. And the results published in the same journal in 2022. The first result on Fullarazirman, the patent is granted to us in 2023, 31st January. This patent application is pending. Hope this too will. Here, different cell lines were used for studies. And the cancer cell lines which were used for these studies include the, CT26, colorectal carcinoma cell line. And these were analyzed using, apoptotic molecules where cell death is programmed using caspase 9 and BAX and cytochrome C in U937 cell lines. CT26 was also studied. And most importantly, 4 T1 breast cancer cell lines, also the results were very good. 4 T1 breast cancer cell lines. Let me announce this, that this was at the concentration of 10 microgram per milliliter or 20 microgram per milliliter at very small amounts. If this works in human beings, eventually it will be a dream come true. What am I saying? Our water-soluble nanocarbon materials, we have proven all the 3 manganese complexes, water-soluble, are showing high anti-cancer activity and very low or acceptable limits of cytotoxicity. Next slide. We spent a lot of money in the next slide. This result is from the Reliance Life Sciences in Mumbai. It's extremely costly to get these tests done. And single-walled carbon nanotube manganese complex showed 75% inhibition. Fullarazirman showed 89% inhibition. And the third one, RGO manganese complex, showed 78% inhibition. And these are spectacular results. I do not know. But most importantly, that these are also working very well against what is called TNBC, triple negative breast cancer cell lines. What are triple negative breast cancer cells? Triple negative breast cancer cannot be detected in the normal manner and is detected very late. By that time, it's not possible to treat the patient at all. Whereas it appears all the 3 materials cost apart. Now we are doing studies on these materials as suggested by the ICMR, Indian Council of Medical Research, in a huge animal facility in Hyderabad, opposite Bharat Biotech. This is called National Animal Resource for Biomedical Research, NARFBR, in Hyderabad. And what we must understand is that this is definitely a very promising area. Next slide. 1 minute, allow me a minute. I need a short break. 1 second, I'm back with you in a short while. Yes, I'm back on the job. Can you hear me again? Hello. This table shows that with single-walled carbon nanotube at 250 microgram per milliliter, there is a 96% inhibition. With the RGO, reduced graphene oxide, it is 95, and also it is 95 with Fullarazirman. Very high anti-cancer activity and the cytotoxicity are relatively low. In the case of RGO, it is minus. That means it's almost not there. The other case is slightly higher, but still within permissible limits. At 250 microgram per milliliter, you can have a water-soluble, injectable drug for treating cancer with little or no toxicity, but very high efficacy and activity against cancer. Next slide. Next slide shows you the similar results here against different cell lines. HeLa cell lines, A549, MCF7. You can read about them. The last but one is the triple negative breast cancer cell line, 72%, 60%, 62% ability to kill with little or permissible cytotoxicity. I thank the organizers for having invited me for the series of lectures. I am not here to advertise my work. I am not here to sell my work. If this is true, I have written in the Journal of Biomedical Nanotechnology that at least my earnings from this work, if any day, I will want the entire work to be tested world over and found to be true. I am prepared to offer my work at no profit, no loss basis. I know how much of suffering there is in a family where a cancer patient is there. So costly and so difficult to treat the patient that the family is almost broke. I am not claiming that we have made anti-cancer drugs, but these are candidates. Should this be true, how many of you are in the audience who want to join our work? We need pharmacists who can make formulations. We need students who can synthesize. We need chemical engineers to tell us how to upscale the whole work, because it's not an ordinary kind of work that you can think of. You have to be very careful. And the different parts of synthesis and then testing biologically against cancer cell lines and then making a formulation and testing in vivo. In vivo means, or in vivo means inside living live animals. Currently, the tests are being done in mice in Hyderabad. I hope good results come. Then we can go to what is called phase 1, phase 2, phase 3. At any stage, the project can fail. How much of money has already been invested on this? Unbelievable. Next slide. We are closing. Almost we are near the end. This is the paper on single-walled carbon nanotube, aryl aziridino carboxylic acid decorated with manganese 2 complex, increased root growth in Arabidopsis thaliana. This is published in Chemistry Select in 2019, December. Next slide. Shows you the work on reduced graphene oxide published in 2022, for which a patent application was filed earlier. Anti-cancer activity of water-soluble reduced graphene oxide aryl aziridino manganese 2 complex. This is on reduced graphene oxide. Next slide. The patent was granted to us, dated, I presume, 31st January 2023. Next. This summarizes the work. Nanocarbon materials on the left-hand side, be it fullerene C60, single-walled carbon nanotube, or for that matter, our reduced graphene oxide. I hope we get graphene also into our hand. In the first step with heat, we heat with the aryl azide. The second step, we do a permanganate mild oxidation. And the tubes, 2 tubes in the middle, you know, represent Fullarazirman. The one on the top represents, I presume, the single-walled carbon nanotube, and the other one is a reduced graphene oxide complex. All these 3 we make in one batch about 80 to 160 or 200 milligrams. That is enough, not enough to even do tests on cancer patients. People have told me that in the case of cancer patients who are terminally sick, if the patient or the family allows, tests can be done straight away to save them. But they must sign and give in writing to the doctor that these tests may be conducted. But a man like me, with a mere PhD, who may be forgotten and misunderstood to be a medical doctor, has no permission to talk about this, because only a medically qualified doctor is allowed to administer. One of our chemistry products from Delhi University, who is Dr. Ganguly, who is maybe, he's around, he's maybe 90, 95 years old, earned a lot of money and donated a lot of money to Delhi University. He made many drugs. One drug he made for treatment of blood pressure. So he went to a doctor in America. Sir, what is it? Sir, I made a new drug and I have a high blood pressure today. Can you prescribe my drug to me? He's an organic chemist. He's a synthetic organic chemist. He cannot take the drug on his own. But if the doctor permits, the patient may take. Same stand I'm taking. I have shown you slides to convince you that all these have high activity against cancer. They are water soluble. They are non-toxic. They will be washed away from the system. They have to be used in very small doses. They are nano. Their effectivity may be very high. Are we through with our slides, young man? Yep. I think the novelty is they are water soluble. They are nano in size, stable at room temperature, very low dose, high anti-cancer activity, low dose, and side effects therefore being less or almost not there. And these can also be useful in material sciences. Yes, there anything more in the other parts of this? We have patents and all I have described. We have publications which I have described to you. And there are young people, 2 people in this audience with whom you can be in touch. Dr. Sujit Kumar Thakur and Manur Arya. Both have been with me. Sujit has been with me for some years. Manur Bhav has been doing tremendous work, not only in this area which has been demonstrated, but also as a student of physics, he is into making nano devices. So let me conclude, but there must be a chance for anybody to ask questions, and I'm ready to listen. But before that, what is the future? I'm not saying the future. What are the areas of the work that are presented where more work is needed? Suppose, as I told one of you this afternoon, somebody asked me somewhere, why did you put manganese? I said, we were doing an oxidation with permanganate. We never expected manganese will form a complex. He said, can't you use cobalt? I said, I don't know whether cobalt can be used. Why not? If you do the oxidation or chromium trioxide as an oxidant, I don't know whether chromium will come. But also the questions, is there a role of manganese? Is there a role of cobalt? Is there a role of chromium? Is there a negative role or is there a positive role? Manganese may be harmless, but may be helpful in various processes in the biological system. It cannot be said of every other element. Another one, you have done a synthesis with fullerene, single-walled carbon nanotube. Can you do this with graphene? We don't have graphene in India being produced. And you can buy it from Sigma Aldrich. How does it help? I do not know. They told me in Kalamaseri they have supported this project with huge amount of money from the government of India. I asked them. They said, 2 saal baat tayar. 2 years later, we can give you graphene. They are asking us questions. Even in the IIT lab, which we are setting up, it has taken us 1 year to get the permissions. This is just around the corner. I was grilled on the question, what is your technology rating level? How close are you to commercialization? Then they have rated us, I think, at about a level of 7 out of 10, which is a high rating. 10 means you are commercialized. Anywhere near 10 means you are in a position to commercialize. I again raised the same point. Is this a work to be done in a college? Most of it, such work was done, Vishwamin was done in college. Where was the lab? What are the size of the lab? What are the funding? Did I only do research? I taught 18 hours in college, 6 in the university, 24 hours. I spent time with the students. I did a lot of administrative work for the college, elections, for that matter, examinations, admissions. But research went on. A college must retain its original structure where we live for each other, not live for oneself. I quote somebody, only those who live for others. Others are more dead than alive. It's a very strong statement, which I also think college is a religious, spiritual organization. We always start, especially in the first year, students start the day with a prayer in the assembly. And it's a very wonderful, I don't know how many such institutions are there in the country where such traditions are there. Those rich traditions and excellence of St. Stephen's College are our targets. Sciences were new to St. Stephen's College, But we have established our presence and we outlasted the dominance of the arts bloc. We were called the sciences. Now I coined in 2011, allow me, sir, anybody who is there, I apologize. I coined the new word artsies. The sciences have actually outclassed the artsies. But we are not competing with each other. Social scientists coming to join our work are equally welcome. Shall we close the session, my friend? And open it to any questions from anybody.
The lecture has concluded. We now invite you to engage in a Q&A session with Professor Eswaran. Please raise your hand or use the chat box to submit your questions.
So there's a question in the chat box.
Yeah.
What is the significance of Multi MS on TB?
What is the significance of?
Multi MS on TB.
No, 1 minute. It's a very vague question. Multi-MS is an instrumental method. It's an instrumental method. TB is a disease. You can't link the two together directly. Let me say diabetes is caused by the lack of insulin. We could only estimate the molecular weight or theoretically calculate the molecular weight of insulin in the past when its chemical structure was established. But when multi-MS matrix-assisted desorption ionization mass spectrometry arrived, with a very small amount of insulin, we were able to derive and determine experimentally its exact molecular weight up to the 4th decimal place or 5th decimal place with such precision. But insulin, it's only the molecular weight. It doesn't cure diabetes just because we know the molecular weight. The question that on the one hand, tuberculosis. What is tuberculosis? It is mainly, Mycobacterium tuberculosis. Mycobacterium means large-sized bacteria with a lot of fatty material like mucic acid and so on. So many materials are there which clog the lung and they actually make the man difficult. It's difficult for the man even to breathe. And they'll throw up and cough up blood and die. So the question should be, how can multi-MS be used to understand the mechanism of action of a very nice drug, a very modern drug? It's called bedaquiline, B-E-D-A-Q-U-I-L-I-N-E. Bedaquiline actually works by in interfering with the ATP-ADP machine. Like in your mother's kitchen or your kitchen, you have a mixie or a grinder. It's a machine. The machine actually goes from one side to the other. Actually, there is physical movement. ATP becomes ADP. Adenosine triphosphate becomes adenosine diphosphate. Phosphate is released in the process. And this is used for various purposes, including controlling acidity in your stomach. I mean, many of us, after eating heavy things at night, report acidity and trouble. It can be treated with the modern drug called omeprazole. Tuberculosis, multi-drug resistant tuberculosis has to be treated over a period of nearly 6 months with what is called triple drug therapy. Yes, if you know the proteins which are involved, and if you know the drug that you are using, you could see how the drug is working, whether it's binding to the protein and the protein complex isolated and its molecular weight determined by multi-MS instrument. Just because you have a glucometer at home, because you have found out the molecular weight of insulin, does not constitute a cure of insulin. So multi-MS does not cure tuberculosis. Somebody said, Panchang, I don't know how many of you know the word Panchang. Panchang is almanac. It predicts what will be the day. It will be a good day or a bad day, etc. Will it rain heavily? So somebody said, take the book and press it. Will water fall from it? That's because it predicted it will rain heavily today. He's a very famous man from Bengal. I'm not naming the man. He was an uneducated man. He gave the simile. The simile is it stays that the almanac on being pressed will not give water. It's only the clouds which will pour water onto us. The prediction is not enough. If you just know that you have an instrument, MALDMS, you can study large molecules, very small amounts, with precise molecular weights being determined. But it won't cure the disease of tuberculosis. You have to understand the disease. You have to develop drugs. You can use computational methods to develop the ideal candidates. Many new drugs have been now discovered, but still multi-drug resistant tuberculosis is a scourge in the country, especially after HIV/AIDS came into the picture. Many AIDS patients are immunocompromised. Therefore, they are candidates who suffer from tuberculosis. Next question. Good question though, but unconnected to the exact topic. Go ahead. There were some people commenting or something I could not read.
Yeah, yes,
Please
sir.
read
So
out.
it's looking at the structure of Fullarazirman in electron microscope, I had the idea of using it from a structural point of view in nanorobotics, utilizing its folded structure as a honeycomb storage or filter, or as an alternative to gecko tape. Tape, what do you think? It's a question from Rohan Kumar.
I have to understand your question before I answer. You said you will use it in robotics.
Nanorobotics.
What is the use?
Utilizing its folded structure as
collect something?
utilizing its folded structure as a honeycomb storage or filter.
Okay. So in the diagram that we had drawn, one word was drug delivery. What you are saying is the same thing. This can be used as a carrier to deliver a drug. Just like I gave this example of ATP, ADP, you can use this machine to deliver drugs. You can use such cages like this. If it traps a drug inside and goes some distance, so many angstroms inside the human body, and delivers it on the other side, it will be a great progress. Well, I look at your suggestion as a hypothetical future development and wish you good luck that you will make a robotic out of it and actually make the robotic walk inside your blood vessel, go into the knees or to the toes, and cure people whose blood supply for one reason or the other is impaired. Next question.
Sir, there is another question from Alok Krishna. He's asking where did Ashok Krishna. He's asking where are you from?
In fact, in return, before I answer, I want to ask him about his background. Where is he from? What institute? What studies he's doing? Then I will answer the question happily. Any answers that he wants. Hello, Ashok Krishna, what are you doing? Where are you from? So if there is a personal touch, then I will answer in a personal manner.
Sir, I am from Kerala and I am doing BSc Honours Chemistry in St. Stephen's.
എവിടെയാ നാട് എവിടെയാ?
Sir, Kottayam.
കോട്ടയമോ? ഞാൻ അയിലൂർ നമ്മാറ എന്റെ ഇടം. എന്റെ സ്ഥലം അയിലൂർ നമ്മാറ. എനിക്ക് കുറച്ച് മലയാളം അറിയാം.
തമിഴ്നാട്ടിലാ?
ഇപ്പൊ കോയമ്പത്തൂർ അല്ല, പാലക്കാട്ടിലെ.
പാലക്കാടോ?
In Kerala.
Oh.
We will meet when I come to Delhi and you will teach me more Malayalam. The only other person with whom I spoke in Malayalam is Dr. Reverend Dr. Valsan Tampu, who resides in Trivandrum and is putting up so many videos. Now I listen to his videos and he's there in Trivandrum. With him in some very formal meetings, occasionally I used to speak in Malayalam when I had to communicate something very quickly and secretly. Ashok Krishna, in the class, which batch, which year?
Sir, first year BSc Honours Chemistry.
Are you at home in St. Stephen's College?
Sir, ipam PG la velile.
How do you like the food in North India as a Malayali?
Sir, ivide Malayali PG appam nammada food ah. South Indian food.
Appo puttu ellam kittum. Puttu.
Puttu, appam, dosha.
Anyway, എന്റെ ലെക്ചർ എത്ര മനസ്സിലായി നിങ്ങൾക്ക് എന്നോട് ലെക്ചർ എത്ര മനസ്സിലായി ഇന്നത്തെ ലെക്ചർ?
സർ എന്റെ ലെവലിനെക്കാളും കുറച്ചുകൂടെ കൂടുതലാണ്, എന്നാലും വരുന്ന ലെക്ചർ കുറച്ചുകൂടെ ഒക്കെ മനസ്സിലാവും.
ஆனா ஒரு 2 சென்டென்ஸ்ல மலையாளத்துல പറയണം, நான் क्वेश्चन கேட்காം, പിന്നെ അത് ഇംഗ്ലീഷിൽ നീ ട്രാൻസ്ലേറ്റ് ചെയ്യണം. இந்த ലെക്ചറോട് സമറിയും 2 സെന്റൻസസിൽ എന്തുവാ മലയാളത്തെ?
എന്തോ
ഒരു സമറി തറ, இந்த ലെക്ചറിന്റെ സമറി 2 സെന്റൻസസിലെ മലയാളത്തില്.
സമ്മറി പറയാവോന്ന് സാർ പറഞ്ഞ ലെക്ചറിന്റെ സമ്മറി തരാവോന്ന്? സാർ ഇന്ന് എന്താ ഏതോ ഒരു എന്തോ സംതിങ് റിലേറ്റഡ് ടു ആന്റി ക്യാൻസർ ഡ്രഗ് ആയിരുന്നു മനസ്സിലായി. പിന്നെ സാർ
ഇത് ഇംഗ്ലീഷിലെ
ബ്രസ്റ്റ് ക്യാൻസറിനെ കുറിച്ചൊക്കെ പറയുന്നത് കേട്ടു.
അശോക് ഇത് ഇംഗ്ലീഷിലെ പറ, ഇംഗ്ലീഷിലെ.
Sir, I heard you were talking something about some anti-cancer drugs, and I also heard something.
Who made it? Who made
no,
this
sir,
drug?
I was a little late to the class.
No, who made it? I have made it. Next question. Ashok, good luck. Meet me in October. Come and meet me personally.
Yes, sir.
Next question, if any.
No, sir. I will be regular to the class.
Yes. Anybody else?
The next.
Anybody else?
Yes, sir. Thanks, sir.
Anybody else?
Yes, sir. There is another question from Rohan Kumar itself. You used computer software and simulation in many times for your chemistry research. Do you have any advice or reference on how to begin in the computer side of chemistry?
Very good. There are 2 teachers, at least in St. Stephen's College. I know Rakhi Thareja is very good in it. Talk to her. There are other temporary teachers who have also done PhD in computer science. Use of computer in chemistry. And so you can talk to them. But many of them, they depend on imported software like Schrodinger software, Gaussian software, which have to be in a dedicated computer. And then if you interact with computational chemists in national labs, colleges, many colleges of Delhi University, and many lecturers who have done PhD in computational chemistry, they will all help you, but they must allow access to that software. And then go into a restaurant and asking, I'm hungry, give me something. The person in the restaurant will ask you, what do you want? I don't know, I'm hungry. That won't do. You should know whether you want rasgulla or you want sambar or you want puttu or you want dosha or you want aloo ka parotha. So similarly, when you go to a computer cannot help you. Computer says, what do you want me to do? You say, I want you to calculate the energy of a nitrine. I want you to calculate the energy of this carbene. I will tell you what can be the possible transition state. I will tell you what is the intermediate. Now you calculate and let me know. So you must go with a specific problem. So you will say how to start. Meet your teacher, Dr. Rakhi Thareja in particular. There may be others who will help you to start. Even in second year, there is a paper, I understand, in second year and possibly also third year, the applications of computers in chemistry. That will give you some starting point. Enough? Next question.
Sir, there are no more questions.
Wonderful. I'm very happy. So how do I thank the organizers? I thank the head of the department. I thank the principal of the college for permitting this series. And we have just concluded the second lecture in the series and hope to continue and complete this series. Grateful thanks to the audience who listened to me with patience. Thank you.
Thank you, Professor Eswaran, sir, for delivering an enlightening and engaging lecture today. Your insightful expertise and passionate presentation have inspired us all. We deeply appreciate the valuable time and effort you invested in sharing your knowledge with us. Thank you, sir, once again.
How well you speak, how well all of you speak. Delighted. Thank you. I'm at home in St. Stephen's College. Good luck and good night. Bye. Can I close?
Sir, there is one more question as
Yeah, please,
you are about to
please.
leave.
Yeah, tell me.
It is from Akansha Kumari Sahu. Can you guide what to do after BSc Chemistry?
I think we need 2 lectures separately, so I'll spend a few minutes. It's a very large question she's asking. So we need to discuss this in detail, but I'll tell you what our students have done. After our college became so well known for chemistry, many of my students have gone abroad, especially to USA, done MS, PhD, and become professors and earned their name. Huh?
Chemistry.
Hello? So many have become professors of chemistry. Some have set up, industrial units. One student went to the North Pole and also to the South Pole, right? There are very few people who have gone to both North Pole and South Pole. Many have become IAS officers. Yesterday, Mr. Ajit Seth, Cabinet Secretary, Government of India, was talking to me from the batch 1970-73. We are very close to each other and we interact with each other. His young grandson will come in December and once again work with us. Many have joined the IAS and civil services. At the same time, many have gone into business, and I'm not against anybody who's gone into business. It is not that you have to do only chemistry. Mr. Jayashankar, the current External Affairs Minister, is from our college. He did BSc. He's External Affairs Minister. Mr. Neeraj Kumar, with whom I was in touch last week, he's in Delhi. He's a chemistry graduate. He retired as the police commissioner, chief commissioner of police, Delhi. So whether in police, in IAS, in the industry, as professors, academics, all areas are open. Some have excelled in sports, football, cricket, and so on. So the range is very, very open. And if you want to remain in science too, it's not only chemistry. It can be nanotechnology, it can be nanobiotechnology, it can be electronics, microelectronics, nanolithography, and it can be so many other areas. For example, areas in physical chemistry and theoretical chemistry, computational chemistry. You don't have to work in an institute. You can be self-employed. What does this? Yes.
Sir, Saptarshi, sir, there are Saptarshi, sir, can you hear me?
I am hearing you very well.
Sir, there are many advantages. Last class I told that being a chemist, sir, we should always look for the upgradation of skills. For example, thermal engineering. This is also very, very important because nowadays we are seeing chemical engineering is also moving up along with the nanotechnology. Chemical engineering, the power plants, we need to say thermo nano composites, that is the chemical recycling, mechanical recycling. These are one of the important things which is going to play a crucial role, because in the upcoming 5 years, what we are seeing is the tremendous improvement in the technology. Because AI, artificial intelligence, has came already in the market, and now we are going to see that. Last day I told about nano robots. So nano robots, then we can say medical nanotechnology. This would be a huge upcoming thing. Is it right, sir?
I mean, who will disagree with you? You are a new young man coming into science in a big way. You are going to explode into science. For example, I did not tell you, single-walled carbon nanotube soluble, if it is actually injected into a cancer cell and you put a laser flash of infrared low intensity, low wavelength, kind of a radiation, a laser or an infrared laser, You can actually hear patakas. Diwali is round the corner. You can hear the killing of the cancer cell, and you can hear the patakas. Actually, this is a very wonderful use of these materials, for which this is called as theranostics, therapy plus diagnostics. And you can not only diagnose but also kill. And you can kill precisely the cancer cell alone and not the normal cell. Your interest in nanotechnology, your interest in nanorobotics makes me predict that 10 years from today, you will be making many things which will be very beneficial to society, to human beings, especially to reduce the suffering of many people suffering from different diseases. So I wish you good luck. We cannot have any disagreement with you. All that you say with so much of enthusiasm encourages me to say, yes, the future of science with young people in St. Stephen's College is indeed very bright. They need only somebody who will show the road. The only way to do is to go to people who have great achievements, meet them, discuss with them, Nobel laureates and others in person. Next question. Thank you, Saptarishi. Next question, if any.
Yes, sir,
I saw some comment.
We have one question from Akansha. How can we work with pharmaceutical companies? Are there any tips? Can we join any pharmaceutical companies after graduation, or we have to do any degree?
I will answer. It's actually, you don't, I mean, there are people who are working in the army at the lower levels. They are the ones who are the fighters. I'm not saying that there should be no soldiers. They retire early and so on. It is a good idea to join the industry only if you have at least a PhD, and preferably a PhD from abroad. Otherwise, somebody will come from abroad, become your boss. He may be or she may be your classmate. You are PhD India, he or she is PhD from America or somewhere. Your own classmate will come and sit on your head as a boss. I'll give an example in a very positive sense, not in a negative sense. I stayed with Tathagata Mukherjee in my last visit to USA in 2011 in his house. Tata Mukherjee came from Asansol to St. Stephen's College, spent 3 years, did very well, then went to IIT Delhi, did MSc, then went for PhD to Cornell, finished his PhD in Cornell University. Then he worked for 2 years in NIH, National Institute of Health, USA. Then he returned to Pune to join a research R&D lab rather than a production unit. See, there are two ways. Industry, you join R&D, or you join the production line. Production line means routine work. You can't do anything new. Whereas R&D, Tathagat Mukherjee works in Pune in one of the brightest firms of India, the best if possible, and he's become very senior man in the industry in that unit, and possibly in the topmost rank, 1 or 2 higher ranks. Tathagata Mukherjee, you can write to him, take his phone number or his email from me. He has joined the industry. He is not a professor. He is earning a very good salary. He's made great discoveries. One of my students joined Glaxo. He's got a US patent, and his patent is already going to be commercialized. He did PhD with me, but he did not join as a lecturer. He did not go abroad. He did MSc, PhD with me. He's doing very well in Glaxo in Bombay, developed a new product for which a US patent has been granted. So industry can be useful, but don't join the industry at a low rank. That means you must be well equipped. You must have credentials like an MSc and a PhD, and preferably a PhD from a well-known American university. Then you get a standing and a respect. Another student, a colleague of your head of the department, is now very successfully, they organized a conference in college this year, I know. He's very successful in the industry. He and his sister were our students in 1990, I presume. And let me say this, he's working for a drug firm, though he's a MSc in physical chemistry. Very often you will find in the drug firm MSc organic chemistry students working. It is not that there are no inorganic drugs. You know Professor Chandra Ray, he is PC Ray. He has set up the industrial chemistry industrial empire in the whole country is even today worshipped. So inorganic materials, organic materials, process technologies, engineering, all come into the picture. My advice is, yes, start in the following manner. I'll give you a suggestion. There is a center which I'm going to visit in Chennai, in Poonamali High Road. I spoke to the chief executive officer, executive director. Her name is Dr. Shreya. Dr. Shreya looks after Malladi R&D or Malladi Pharmaceuticals. Malladi Pharmaceuticals. She will take you as an intern. Go there for 1 month, 2 months. They are producing. They are not doing so much of R&D. They are producing drugs which are in the market and bring money. Dr. Shreya Malladi Pharmaceuticals. Dr. Bharat Tandon, 1970-73 batch, is running what is called, he works on silk proteins, and it was wet care. Now it is seri care in Bangalore. You go, spend a month with him. But he's an industrial unit. He works on silk. There are other students of ours who are in the industry. There is a person in IICT, Indian Institute of Chemical Technology in Hyderabad, who is 1 year younger to me. He's still an advisor with this CSIR lab. He's produced so many drugs. I invited him to a meeting in Delhi, and the man who was financing the meeting asked me in Hindi, भाई आंख नहीं नजर आ रहा, कितना वो ड्रग कितना बेचा उन्होंने? मैं कहा सर $150 million का बेचा है. ये वाला ड्रग कितना है? $350 million बेचा है. He is 1 year younger to me, Dr. B. Gopalan, care of IICT laboratory, CSIR laboratory in Hyderabad. There are many other examples abroad as well. People, if you write to BASF, Germany, Bayer, Germany, and DuPont, America, Pfizer, America, they will allow you to come, but obviously they won't tell all your secrets. Do you go to your neighbor's home and tell about all the secrets of your home? So no pharmaceutical company will allow you to steal their secrets, but they will allow you to see. Malladi Pharmaceuticals, I've spoken to her, such a wonderful person. She possibly is belonging to the Malladi family who own it. She is a chemical engineer and she is executive director. I'm going to visit their unit myself very soon. Is this a reasonable answer? Otherwise, meet me. We can discuss this question in detail.
We have another question, sir, from Tamanna. Sir, I am pursuing BSc Honours Biochemistry. Want to work on drug designing or gene transfer. Please guide me for further studies in foreign.
Foreign? Why not India first? You are in BSc, na? In which class are you in, Tamanna?
Sir, I am pursuing Biochemistry Honours from Daulatram College. I am in second year.
अभी तो बहुत लंबा रास्ता है भाई, बहुत लंबा रास्ता है. जल्दबाजी ना करो. सुनो हमारे यहाँ दिल्ली यूनिवर्सिटी में ही 2 कॉलेजेस हैं. खास एक कॉलेज है जहाँ बायोमेडिसिन टीच किया जाता है, राजगुरु कॉलेज ऑफ अप्लाइड साइंसेस, और एक पूसा इंस्टीट्यूट में है. और हमारा लखनऊ का लेबोरेटरी सेंट्रल ड्रग रिसर्च इंस्टीट्यूट बहुत फेमस है. बहुत सारे लेबोरेटरीज इंडिया में हैं. अभी दिसंबर आएगा तो दे विल अलाउ यू टू अप्लाई फॉर Indian Academy of Science, internship. Let this information come to you through resonance. You can apply, say, I want to come and spend 2 months with you. They will give you travel. They will give you payment for 2 months and allow you to work with some industrial unit or drug design unit, drug discovery unit. There is this next door to St. Stephen's College. On the back gate, you go to the Ambedkar Center. Ambedkar Center. There are many scientists working, and they're very well equipped. For that matter, South Campus, there are people who are working on your area. Jawaharlal Nehru University, IIT Delhi, Housecast, so many other institutes. Particularly, there is an institute which is in Mohali, more than one institute. This is a NIPER, Mohali. They're working on drug research. Write to the director, go and personally meet, and even get a fellowship from the Indian Academy of Sciences and work on drug design. There are umpteen number of places in India where, for example, Reddy Labs, they will take you as an intern. Apply to Reddy Labs, Hyderabad. See, I want to work with you. There are Sun Pharmaceuticals. There are many, many areas and many, many institutes which are doing tremendous work. But remember, there is one problem in India. Nobody wants to discover a new drug because it requires a lot of investment of money. Shreya told me, we don't do any new drug. We don't try to discover new chemical entities because it will take 15 years to develop. The regulations are very strong. You have to study toxicity. You have to see population studies. You have to see so many things before a drug is approved, except in the case of COVID-19, very quick emergency approvals are given. So you will learn in the industry mostly known drugs and their method of production, their biochemistry. Huge number of possibilities on which we can be in touch with each other. I'm prepared to guide you, tell you whom all to contact. Yes, your question now, where to do it abroad? Oh, the Britishers are very good. The Americans are very good. The Japanese are too good in everything that they do. Therefore, I suggest that you think of, if you can think of, writing to these organizations, Germany, America, look at especially, industrial units. But how do you climb onto a third floor if there is no lift? Do you fly and go to the third floor? Tell me. Answer.
Sir, is
भाई थर्ड फ्लोर को कैसे जाते हैं बताइए कोई शॉर्टकट नहीं है there is no shortcut so you have to go to the second year third year then do an MSc then do a PhD then do good work but you can go to Tathagat Mukherjee sir Dr. Ishwan spoke about you why won't you like a daughter take me into your unit officially I will come and spend 2 months with you तथागत के पैर मत छुओ भैया छोड़ना मत तथागत के पैर पकड़ लो क्या डॉक्टर साहब ने बोला डॉक्टर ईश्वर ने बोला आपको मेरे को ट्रेन करना ही है इंडिया में पुणे में हो जाएगा ट्रेनिंग और लोग उसको अच्छा माने जवाब मिला नहीं मिला
Yes, sir. Sir, like we have, like in America, they are asking a 4-year bachelor's course. So is it compulsory to?
This is a question that has been asked for many, many years. Many Stephanians have been allowed. Many Stephanians have been allowed only after just a 3-year graduation. But that's not a big problem here. You can join for an MSc and do your GRE and so on. But by the time 1 year is over, then you will say, why not get an MSc? Finish the MSc को जल्दी थोड़ी कि अमेरिका जाना है अमेरिका बाद में भी जा सकते हो बोलिए अभी आपकी उम्र अंदाजा 20 साल की है 19 साल 20 साल की होगी MSc होगा तो 2 साल और ऐड कर दिए PhD होगा तो you can add another 4 years you are about 26 or so that's the problem in India people are not getting jobs even at 34 and when they go abroad they don't get a professorship immediately and if you are beyond 35 in America they don't give you a tenure track position you become a professor very early in America and that is you should be good you should have shown good work so you don't have to only go abroad I'm not discouraging you you can go after third year you say वो तो 14 साल ने मतलब extra 14th year नहीं है training कितना year बोला था आपने that is
4 years sir 4 years sir.
वो 4 years नहीं है 4 years 1 साल यहाँ लगा लो ना कहीं और कहीं आपने प्रोजेक्ट वर्क किया तो उसकी रेटिंग बहुत हाई होती है अमेरिका में हमारे कॉलेज में ही इवन कंप्यूटर असिस्टेंट कैलकुलेशंस करके वो पेपर छापो उनको वो बहुत कंसिडर करते हैं रेटिंग बहुत हाई होता है अमेरिका में so I would suggest finish your BSc honors from Dawlat Ram and then do an MSc from some good place in India say ISC Bangalore or even Delhi University or wherever you get admission and then you may go for either a master's or a PhD abroad so you don't wait and depend on your father or your mother giving money to you you become independent soon after MSc or during MSc I mean I didn't have to take a pie after my MSc previous I got a scholarship and that money was enough for even my MSc. And the moment I finished MSc, I got a PhD fellowship. The moment I got a PhD fellowship at the age of 21 and a half, St. Stephen's sucked me. And I worked only in St. Stephen's, but I went abroad to all places that you can imagine, doing science, not as a mere tourist. तो मैं आपको encourage करूँगा, patient रहो. Don't try to climb to the third floor straight away. You have to go step-wise manner. There is a lift, you may go in a half a minute. But if there is no lift, you have to climb step by step. Good answer or a bad answer?
Yes, sir. Good answer.
चलो और बताओ, कोई है? अच्छा लगा इतना सवाल, हम आंसर क्वेश्चन आंसर बहुत अच्छा लगा. मेरे को अपना टॉक से ज्यादा मैंने इसको ज्यादा एंजॉय किया. हम इसी तरह सेंट स्टीफेंस में काम करते थे. हमारे पास बहुत वक्त था, कोई जल्दबाजी हमें होती नहीं थी. बताइए और हैं?
Yes, sir. Akansha is asking what is the surname of Shreya from Maladi Pharmaceutical.
I don't know. I know her only Shreya. I was introduced to her. I spoke about 3 weeks back. She was just amazed at my talk. And she said, unbelievable. We don't do anything new. We produce. And you see them on the internet. I am amazed the way they have production units and so on for known drugs for which they are in the market. And nobody runs an industry without profit. They must be making huge profit. And if you have such an experience, even suppose you don't do an MSc, you spend 1 year with them and they give you a very good certificate. Americans will love it. Americans will simply love it. किसने कहा कि MSc में जाके 1 year करो? वहां MS कर लेना और यहाँ 1 साल ऐसा कोई फार्मास्यूटिकल इंडस्ट्री में हो, तथागत मुखर्जी का असिस्टेंट बन जाओ, श्रेया का असिस्टेंट बन जाओ 1 साल के लिए. वो आपको सर्टिफिकेट देगी. Yes, I have seen such and such person, so and so Akansha. She worked with us on this, and blah, blah, blah. And she is brilliant. And I recommend, or we recommend her for a position or a scholarship in America for doing PhD. कोई रोक देगा आपका? किसी ने ये तो नहीं कहा 4th year should be only doing an MSc. किसने कहा है? आकांक्षा बताओ, जवाब अच्छा है नहीं है? बताओ.
Yes, sir.
1 साल या तथागत के साथ पैर पकड़ लो, श्रेया के पैर पकड़ लो. हमारे जानने वाले लोग हैं. तथागत तो हमारा स्टूडेंट है डिपार्टमेंट का. श्रेया को मैं रिसेंटली इंट्रोड्यूस हुआ हूँ, बहुत अच्छा काम कर रही है. और उनका सर्टिफिकेट 1 साल का मिल जाए तो कौन सी MSc की प्रीवियस की जरूरत है? 4th year नहीं जरूरत. Any other questions? I'm delighted. Please.
There's Sir, Akansha is asking how she can connect with her. Do you have any email or something?
With me?
Yes, sir.
My email, I announced to everybody, my email at the moment is SV, S for Shimla, B for Victory, small letters, SV dot, my name, ESWARAN, @gmail.com. I see all my mail every night, even on the mobile. We can talk on the WhatsApp.
Thank you, sir.
Yeah. Any other comments? Saptarishi is still asking something. What is it, Saptarishi?
Saptarishi is asking that I am in MTech Nanotechnology. I want to do PhD in Australia. I have done 36 publications. Can you guide me, sir?
How many publications?
36.
That's a huge number. I'm amazed. The Australians will simply grab you. There are so many universities. Queensland's university, I have been associated in one manner or the other with Deakin University. So you can write to these universities. They are looking for good people. And many Indian professors are working there. 36 papers is hard to believe. If it's the case, it's brilliant. People will simply grab you. Send me through any one of you or directly your list of your publications. I will see what is on what areas. People will simply be looking for such people all over, not only Australia. Everywhere in the world, Indian students are very welcome. They know they are hardworking. As President Obama said, they are very good in mathematics. They are very good in computer science. In the past, they would sleep on the lab benches. They would eat very little. They don't get drunk like Americans. I hope what I'm saying is true and not wrong with modern students. Good luck, Saptarishi. Deakin University, Queensland University, New South Wales. There are so many. Are we near the end of the question answer session? Which I enjoyed much more than even my talk. बताइए. कोई है और?
I think, sir, there are no more questions.
Yes, let us slowly bring it to a close. Shall I close now?
Yes, sir. There are no more questions. So we can.
Yep. That is the conclusion of the day then.
Yes, sir. I guess so. There are no more questions from the audience. We can conclude the day 2 of our lecture series by Professor Eswaran. And we thank everyone who has joined us. Thank you so much. Have a good evening.
Very kind. Delighted. Till we meet again soon. Bye.